seeing you, all of you, and thank you for coming. And I'm truly thankful, Dr. Priya, for your for bring me here in this campus. Because every year I get connected with your students and they do miraculous job in my lab. So some of the things that I will be talking, actually, they have been part of it. I have been connected with Dr. Jingre. Um, I have been part of IWSA's program wherever it was felt, and the different level of things that just the way it's presented already to you. So at different spheres where women support is required, because the you know in our master's class or any college class, if you see, at least 50% are women who come but then in the career front, we see it is still 10-15% at the max, 20% in the staff strength. So somewhere uh, there are lacunes, and that is where the society needs to look at. And this is the job that IWSA is doing meticulously. And it is really, I feel really honored whenever I get connected with such efforts. So thank you as well. Uh, so coming to the talk, I was told that, if you can connect with the topic of nanomedicine, and this is a field that some of you would know that it's doing miraculous in colleges. Nanomedicine, nanotechnology, nanobiotechnology related research is not rare. So you know quite a bit. Just to remind you, it do not have a it is not even 100 years history for this field. 1969, it was 1959. So that was the father of nanotechnology, if you say, Richard Feynman. So that's the concept came that the same material that you see, like सोना चांदी सबके घर पे होते हैं, but that when you bring it to the molecular level, that gold is do not glitter anymore, so it looks like a black particle. So it's a new feature that comes in, and how to use those new features, and that is where is fueling this field of research. So I do very little. I am still a biologist. I'm not a material chemist. But I like holding hands with others in the field. So that comes in me naturally, because I have grown up in my research career. I grown up in imaging lab where chemists, physicists, engineers, biologists, and clinicians, we all have worked together for years to make this medical imaging field grow and support our vulnerable disease area. So since I came here in ACTRIC, I'm now more focused, only focused in cancer, and particularly I have chosen the breast cancer as a major field for me, for my research. So I will touch upon this, but this being a popular lecture, um, I have brought some of the things that probably it's not very vivid in your course work. So it will still be something little bit new. And since this is something, I have only 25 slides, so I'm giving you the plan. So, feel free to interact, because some of the things I'm only taking bits and pieces and try to connect with you. Okay, so that being said, uh, So let us start with cancer, since I'm coming from AgTech and it's nothing unknown. You have a cancer biology in your course. So I like to start with this very simple video. And there is no word required. What you see, you can explain me that yes, the cancer, a single cell, it has started, uh, it has grown, it has proliferated, then it draw blood vessels to get it supplied, and then it further grow. And what we do in the end part of the video, what we try to see is in the research lab, what we like to see, that it shrinks. And we try to find out ways to shrink it. And this is where I will connect you in the latter part of my talk with the nanobiotechnology, what are we doing and how we can do that. So just to reiterate, this is well within your course. So it's the leading cause of cancer. So still the leading cause of death other than cardiac disease. So this is one of the prime disease that kills people across the world. And so it's a great concern. And but in all cases, it starts from one cell. That also we know the biology, the way we understand today. So you can see that there are, you read about these oncogenes and overexpression of oncogenes. Sometimes when proto-oncogene get activated as an oncogene and trigger the cancer onset, and also the loss of tumor function, suppressor gene function, either by mutation or by deletion. These tumor function suppressor genes, when get inactivated, cancer can also trigger. So, so there is the involvement of mutations. There are involvements of various kinds of mutations, and which happens because of various source of triggers that is in our environment, in our culture, and all sorts of forms it can come, starting from food as well. And then the in the middle, what I have given shown you is the uh, actually for the online. Let me take out the laser. So these are the features that comes to your mind readily when it comes when one asks you that how do you understand cancer? So it's an uncontrolled proliferation of cell which do not know how to stop. So it lacks the programmed cell death feature. So it has suppressed that feature. Contact inhibition without any basal support, it can grow, and then it starts draw the blood circulation so that its energy supply and excretion can also be supported. So that is the angiogenesis, which is the new blood vessel formation, and then activating invasion and migration, which is in the advanced stage. So where cancer cells can spread out from one spot where it started, and then it could be a more devastating state and goes to critical organs, and then it can be thing. So now the whole thing is shown here in a human body, and you have to imagine that 100 years before, how doctors could realize that there is a cancer. If it is on the surface of the body, of course, it's palpable, it's either visible or palpable. So that way, it can be detected in an early stage. But because the body is opaque, so how really, if it is inside any of the organ, how really we reveal? And, NCI had a call given in around 2000 after post-genomic era, that war against cancer, because NCI director Harold Varmus gave this call that, we need to declare an war against cancer. So in war, you go all out against your enemy. So consider cancer as an enemy and then fight against it, bring the best that you can, and with full force. So, and then the whole research field got fueled in tackling the adversities that cancer poses. So ultimately, what happened is that we realized that what is required is late stage disease we can handle to some extent, but we cannot cure it. We cannot flip it back because it has advanced so much. It is very complicated, and that's where major challenges are. So the field realized that what is required is an early stage, and early detection of cancer is the something that was taught. And this is where this diagnostic improvement of disease diagnosis becomes so much important, because when it is a deep-seated organ, how we really reveal in an early stage, because why, that I will explain you in a later slide. So let's move ahead. Staying connected with your course, so you know very well that so a requirement of visibility is required, and at the cell level visualization. So it all started with the microscope, and as you can go as back as Galileo's invention of the compound microscope, which then the first live cell was observed, Leeuwenhoek. So you all have read in your 8th standard, 9th standard, 10th standard books. Uh, so over the period of time, so this evolution of microscope continue to happen and give us various ways of looking into cells and revealing the features of cells. Its cell itself is a complex, like a body, you can say, where multiple ultrastructures and their coherent activity actually make the cell function, right? So, so going deeper, so the transmission electron was actually the very good invention that fueled and that helped to go deeper inside the cell. So people started looking at the mitochondria and so on, and where a lot of research on today is focused on the mitochondria. Mitochondria is a unique ultrastructure in the cell component, which is always comes from your mother. So it's a maternal inheritance. And so it has its own DNA. So it's a very intrigued biology that is involved in the mitochondria itself. So anyway, the field has moved with by gaining different kind of microscopic inventions. The field continued to move, but one microscope that is not in my list on this slide is scanning tunneling microscope. It was invented in 1981, and Jared Binning was one of the scientists, along with another scientist from Germany. They invented this scanning tunneling microscope. And that was the first thing that helps you to see at the atomic level. And then this microscope actually, 1986, the newer version of it, that is the atomic force microscope, which is today a lot of people are using, and it gives enormous power in understanding the biophysical, biomechanical properties of macromolecules. So, and that journey is continuing. So there are other different microscopes giving us watching cells and live. So beautiful videos comes out, and sometimes you might have encountered in your search in internet as well, right? But that goes to the cell history and the journey, how we can visualize cells. On the other end, the medical fraternity, so they started actually looking. So okay, none of you are seeing the title. Uh, uh, the the other way, the medical fraternity, for them, it's going down to cell. It's tough, when a person comes up with some problem, one has to understand what is going on at the tissue scale. So there is a huge scale difference from micron size or even lower than that, nanometer size, Armstrong size, um, to coming to a centimeter inch resolution. It's a huge challenge. So that was the thing that was okay. Um, that was the thing that started looking into, and the field has progressed around, you can say, 1900, when this radiological diagnosis came, the X-ray, and that started giving the contrast development between the solid, like bone tissues, hard tissues versus soft tissues like muscles and so on. And then at least if there is a spread in the structure of bone, or any deformity that is happening in the bone structure, that could have been then started visualizing through X-ray based thing. So the field continues to develop, and then there are a couple of other modalities that added in, and which use radioactive material. And my next slide, actually, so you might have already gone through this. So there are also other radiological tools like sound-based imaging, which is very safe because it's not radioactive, it's not photon. So even photonic signal also can have toxicity in certain tissues. Um, so in that sense, sound is the safest, and that's why pregnancy test is always done using ultrasound, right? And then the higher version of X-ray imaging, that is the computed tomography, which you all know of in hospital. It is all hospital has a CT scan provision. MRI is still rare. The magnetic resonance imaging, it also get to see the body structures internally in a whole body scenario. And then the positron tomography, which actually fueled this field where I'm stuck at, and a grown fascination, that is the molecular imaging, which where we are talking about watching molecule working in the live body. So that is what we wanted to achieve. And here I categorize this whole set of imaging power that we have gained into two categories. One is a anatomical imaging, which basically help you to see the structural change, and versus the functional imaging, which helps you to see when the molecule is working in a work. So the function. And coming to this functional imaging part, this is an intrigue story for me all time. So I thought I will also share with you. Some of you might have heard this name, George Charles D. Hevesy. And he was the name who actually changed the field. And this cartoon shows that his madness. And many times scientists are mad. You all know that many inventions are serendipitous. Sometimes sheer madness is involved. So this is the case that he was in a PG. So he was a scientist, and the landlady, he has a suspicion that the landlady is bringing back my leftover food in the next day and serving me. And so what, he was a radiation scientist. So he had possession of radioactive iodine, and so which can come in a salt form. So after he finished his food, leftover food, he sprinkled the iodinated salt, which is radioactive. And next day, first he scanned. If it beeps, then he will not eat. He will not go ahead. So what it showed, it's a sheer madness. It's a suspicion, but essentially it brings out a scientific principle in front of you, that how radioactive materials can be used to traverse over time. So the unseen, which is the night phase, perhaps, what has happened to that leftover food in the night when I was not present? Now, next day, you can actually track and confirm and then go ahead. So this principle actually fueled the field of nuclear medicine, where single photon emission tomography, positron emission tomography later on came up. Okay. So anyway, his other madnesses are also here, in Nazi time. Uh, Even the Nobel gold coin. Nazis used to snatch from the scientists. And he actually dissolved that gold coin in a if you put it in the nitric acid, it will melt. So he made, to save it from Nazi, he didn't even bother to dissolve his Nobel medal in acid. He can recover it. He recovered it later, post-Nazi era. So these are the story you can read more. So he was a scientist of that caliber and depth, chemist who more focused on radioactive chemistry. This slide I just took up because these are not something that you do. So there are many forms of radioactivity and which can be used for diagnosis these days. And the single photon emission, what I said, the gamma beta based emitters. And then the positron is a very specialized nuclear medicine tool, diagnostic tool that actually use a positron, which is a electron, it's a negative charge, but positron is a positively charged electron. And this is generally very high energy. So when from an unstable molecule, the positron comes up and it collides with a nearby electron for the next molecule that is nearby. So on its electron band, when it collides, then there is an opposite direction, 180 degree, to emission comes. So these become an added advantage over gamma camera, which is being a technology principle for SPECT imaging, where the unstable molecules emits single rays in all directions. So finding a coordinate is always an important and gain because it helps you to find where the source is. And that is what positron gave, and that's where positron could improve the quality of image that you can get with a more clarity and up to 2 millimeter by theory, but in practice, up to 5 millimeter, it can predict the right tumor focus very well. Just another slide on the diagnostic. So this is also in your physics principle, you might have read that the wavelength and energy, how they are inversely correlated, and higher the energy, lower the wavelength, and like red light has less energy than UV light, right? And and in this whole range, so light that we sense is only this much. It's a very narrow domain that we can observe, our retina can catch, but there are many other different rays in our vicinity all the time. And starting from gamma rays to X-rays to ultraviolet, and on the far side, it is the infrared, radio waves, and so on. So here the energy is less, wavelength is high, and they are safe because they are less energy. So they break your DNA laser. So when we are getting exposed, so that's why ultrasound is a more safer than a X-ray based scan because that is a higher energy and that can split our DNA, which can Of course, we all have the ability to repair our DNA, that is also integrated. But sometimes if it is misread or misdone, then the chance is that the mutation has arise. Okay. So one of the area that I will focus primarily for biologists, it is good enough where we are not dealing with patient, we are dealing primarily with little mouse, which are our patient, and we do experiments by modeling on them. And for this part, this photonic signature is safe, it is non-radioactive, non-hazardous, and there are ample scope that we see, and some of that I will just touch upon quickly. Um, uh, is that everything all right? Okay. Coming to the photonic based imaging, so there are primarily 2 or 3 different mode. One is the fluorescence, and some of these blue range, UV range do not work very well in tissue because that's where the hemoglobin absorbs the light maximally. So it gets suppressed. So anything above 600 nanometer, where 600 is, so around 580 onward, it's the red. So red, far red, infrared, these are the imaging which works very well in a tissue scale. And there are also another class of protein or even other molecules, chemical molecules as well, which emits luminescence signal. And this, how luminescence is different than fluorescence. Fluorescence require an excitation, and then the molecule goes to the excited state. Very quickly, it releases some of the energy and then slowly transit. And while this transiting, so that's the emission, and this is the excitation. So there is you have to hit with the photons of higher energy, and then you always get a lower energy photon. So if you have a green fluorescent protein, you have to trigger it with blue light and you get green. So that is again on the red side, so lower energy. When it is a red fluorescent protein, you hit with a green light and you get the red fluorescence. So the loss of energy, that has to be balanced, and that's where the emission that comes out is different. There are also multiple fluorophores, very synthetic, very exciting area, and starting from quantum dot to all kinds of dyes, even natural dyes. So there are a lot of these fluorescents that are around, and which we are also very well integrated in our imaging system. But all of them require the light excitation. So that's where the other class was very, very important. So, but before going to that, so this is just to show you the example that there are plenty of options that chemists have added in terms of developing fluorescent dye. The biologist has also added options. So something like the 2008 Nobel Prize winners. Um, so Roger Chen's and Osamu Shimura was the first inventor of GFP. And then this GFP actually went in the hand of 2 others. And Roger took this effort of doing mutagenesis. So it is sort of a playing over God's creation in a sense. So, it's a natural GFP protein, which is in coral, and then getting that DNA, and now you analyze rationally, where do I do the mutation, and then that mutation will change its color, change its emission, quantum output, stability, all kinds of playoff you can do. And Roger was actually did that and brought this whole slew of color variants out of the GFP. He backshifted, which is GFP towards citrine. There is something called citrine, which is slightly bluer side, and then all the way up to cherry. And he named all these in the fruit variant name. So banana, cherry, citrus, orange, so on and so on. And all these are commercialized today. So these are added very good options for biologists for doing genetic experiments. that helps you to, you can tag one gene with cherry, another gene with orange, another gene with banana or GFP. And then just by looking at the color, you can say that I am looking at candidate A versus candidate B versus candidate C in your assay. So this has opened up a whole option for you to perform high throughput screening, where this kind of spectral separation can be easily done. And looking at the spectral character, you can say what you are, which molecule you are actually looking, because that you have controlled in your through your experiment by cloning together as a fusion gene. The other set of genes that do not require you to excite with the light, and they are basically chemical reaction. It's an enzyme that are something like firefly, where so this gene is also very popularly used in biology sector. And so this enzyme can catabolize a substrate called luciferin and produce a very specific light. So now how it is any different? Here it is the light coming out from dark, absolute dark, because it is a chemical reaction that is producing light out of nothing, right? So there is no blooding, there is no requirement of phototoxicity, and they are very dim light, but because they are coming from darkness, they can be very well sensed. So shown here are some of the cells which are positively selected, engineered for firefly luciferase added with the substrate. This becomes a great tool for us because in the mouse laboratory, where we test disease specific like cancer, the growth of cancer over days, without killing that mouse. So the same mouse can be brought back every other day, and they are very non-toxic reporters, so they don't harm anything. All you need to do on the day of imaging is inject the substrate, and in 10 minutes time there would be a flash of light from the source, because this is also controlled because it is ATP is a cofactor. So if the cell is dead, you won't find the ATP in there, and therefore no light comes up. So essentially when you see a light coming up, you can say that it's a viable cell growing. And so the number of photons in one place is linked to the cell growth. Because the cancer, we know the property is uncontrolled proliferation of death. So you can see that within 16 days, a few cells that we implanted become how many cells and how big a mass of photons that is coming out from there. And further down, it is spreading elsewhere, like other organs it is spreading. Final day, you can take out all the organs. So something like this, we call ex vivo imaging. This is we called in vivo imaging. So ex vivo, you can organ harvest, and then you can reconfirm that, like brain with metastatic nodules, lung with metastatic nodules, liver with metastatic nodules, and so on. Okay. So this brings me back to I may have another 15, 20 minutes. Okay. Uh, so this brings me back to refer to the very second or third slide that I was talking, the biology, where we have to understand the cancer, which way we want to understand the cancer, and which way would be the best for us to diagnose the cancer using modalities that is available. So this is where the call should be. So looking at the structure. So what are the modalities? So something like CT, something like MRI. So these are the scans which reveals the atlas. Okay. So it looks at the structure. So now it's the medical doctor, the radiologist who look at your scan result. How do they detect? Or how it can be missed. If the doctor's experience is matters, or the state of mind when he's visualizing the scan, will matter, because it's a comparison. So while he's looking at the film, he's co-registering it, how whether it is matching with his atlas that is in his mind. So that's where physical, there is a chance that, and that's why scan results are never 90% accurate. They're generally 75-80% accuracy is there. MR has, of course, improved, and there are other ways of improvement in all of these modality, like using contrast agent and so on, so that that also helped improve. But it is still not 90%. So there could be always 10% chance that a disease that was there, that was missed, and so it has got an escape to grow further, and then perhaps at latter point of time, because now it is more obvious, so it would be treated. But then it is a late stage. So that's where it may add a chance to fail the early detection that was voted at that point. While the the other modality like PET or SPECT, what they can do, particularly PET, is a functional change. So all of these, so there are different probes. So proliferation, you can link, this is where your biology mind comes in place. So what should be the molecule that we should connect in context of proliferation measurement? Uridine, thymidine. So these are the basic blocks of DNA, and anytime a cell has to divide, new requirement of DNA synthesis. So this is where would be requirement. So when the cells are highly proliferating, obviously they are in need of these a lot. And now if you can tag F18 molecule or a positron emitting molecule, attach with them, then it will be accumulating continuously in there. And that is what will help you to see where they are accumulating. And so you know that, yeah, this could be the spot where the cancer is growing. It could be the TB also, because that is also required of that sort. So that could be, if a TB patient also have lung, the cancer is there, then it could be confusing, and there should be other tests to be done. Okay. So I leave it there. So this is basically, I try to explain you that we have to weigh in that what should be the right thing to do at what stage. So if it is early detection, functional assessment, because there is, the molecule that has changed because it is, the disease started in one cell. Now, identifying one cell in a tissue scale, perhaps an extreme challenge. We are nowhere there today. But we can, at the max, we can push like 2 million or 5 million cells in one spot. We can do that. We have that ability. So that is like 2 millimeter, 5 millimeter of a tumor. What CT-MR does, so most of the time glioblastoma, 3.5 centimeter tumor. And that's the first time the symptom came, either something happened, person was hospitalized, CT scan was done, found, yeah, there is a 3 centimeter tumor, 3.5 centimeter. So it is a big mass already grown. So the mass brings, when it is enough grown, only then there is a symptom. And that is what is the challenge for just depending on the structure. In India, we don't have choice. We are still going with structural scans. But in the US and all, the positron emission tomography is covered by insurance, at least one annual thing. So it helps the whole body assessment for, the FDG scan can be done. Now I am leaving the diagnosis domain. We will apply the diagnosis in our mouse models. But the arena that we want to focus in the context of nanomedicine is the cancer therapy. And what we see that we have history of development with these three primary hope for us when a person is detected with cancer. So either surgery, radiotherapy, or chemotherapy, or a combination of that. Okay. But, you know, there are a lot of refinements that has improved these, but still what is very prominent on today, other than all the difficulties that are mentioned here, is now there are more and more evidence that comes that tells that all of these processes actually help release cancer cells in the blood, and that's where cancer bounces back. So there may be a temporary cure, which is up to 3 years, 5 years of life gained, but then cancer will come back. In certain disease like breast cancer, there are survivors who are 20 years. Survival has been ensured for certain types of breast cancer, not all types. But remaining, there are more aggressive types where these are still vulnerable. Other types like glioblastoma, once detected, expected lifetime is 1 year, at the max 1.5 year. Pancreatic cancer also similarly like this. So there are challenges, and um, so it's not the ultimate thing that one can think, and perhaps for the new generation, we have to think even harder that what should be the ideal cancer treatment. And this is where there are many theories. If you will read as you grow in your studies. So there are theories. So something like called inherent drug resistant cancer. So where the cancer cells are very heterogeneous in nature, and in a mass there could be a different set of cells which are different than the major mass, and the treatment is for this mass or that mass. So both mass cannot be cured through one treatment. So naturally, some remains and that grow back. And this is one concept. While the other concept is that the chemo or the radiation exposure, this is also very much vivid in the research literature nowadays. Our group has also worked on radiation, and we see that even radiation therapy also means there are escapers, cells that escape and then they grow back. We call it radiation resistant cells. And so they are very plastic. So whenever such assault is given to treat cancer, the cancer cells are very smart. Their internal signaling network changes, alters. They manage to escape and so this is called acquired drug resistant or acquired resistance, where because of the assault that they were exposed to, some of them managed to convert into a different class, and then they bounce back. So there is a very fine difference. And now there are different terminologies people use. Some say cancer stem cells. So you can say these are cancer stem cells which were quiescent, because of which they were not very exposed to the treatment and so on. Okay, makes sense. Okay. So that is where I bring this analogy that, this is like a python, and means you are trying to control cancer, and cancer is pushing back and giving the pressure back on your hand to get released and escape. So the fight is on, and um, so young minds are required very much to think in a new perspective, bring your new vision. What should we do that can really make a change in cancer patients' life, even better than what we can achieve today? One such thing was very simple, and I connected it very well because I always liked light was the most favorite chapter for me in physics course. So I connected very well in imaging, and I also then connected here. So There are many molecules in place, like gold, I was telling. It is no more glittering. It is like a black particle to me. It absorbs at 515, 20 nanometer range. Um, but we can do miraculous things because gold is the outer band, quantum band is the heavy electron band. So, so these electron band oscillation, when you trigger with a photonic signature where the material is tuned, then it oscillates. And the oscillation of its outer band triggers uh, uh, the temperature. So again, it is something like energy conversion, you can say. You are triggering with photon, but it is releasing heat. Okay. So something like, all of us have done at some point of time in childhood that with a magnifying glass, you can bring sun and burn the paper, write your best friend's name or your own name or something like that, right? So similar kind of concept that, in the blank paper, if I trigger that laser, nothing happens. On a water spotted place, nothing happens. But my particle, nanoparticle, gold nanoparticle, which are black with mark, and I trigger it with the laser and it burns out. You can see the smoke coming. So, so in a plate of cells, the cells are here stained with red dye. So a monolayer of cells added with the particle and triggered with the laser beam. The beam is 5 millimeter beam, so it do not cover the whole well. So you can see the beam area, the cell immediately vanish. So this was a very intriguing and shocking sort of a result to me in the beginning run. And since then, I fall love in this therapy, and I am continuing pushing it, and with an expectation that at some day I'll be able to find its integration in the cancer clinic because it does miracle. We saw that just the particle, it's very biocompatible. It do not kill cells. Toxicity is very minimal. But the moment I add with the laser, the cells die, and 15 to 20% cells can hardly survive within an optimized. And it is across multiple different types of cells. Even we have tried different other therapy resistant cells like chemo resistant cells, radiation resistant cells. So they all, we can, because this is a physical therapy, the cells, why is the cells die? Because of this, that, the, we all suffer with fever, and we know that why mom will not OD if the temperature is very high. And this very high is something like 47 degree, 103, 104, and that is really threatening. Beyond this, if it is 105, it will cause severe brain damage. So what is the effect of thermia on our cell? Because we are homeothermic, we are a normal body function at 37 degree, but if it exceeds beyond 10 degree more, it's a absolute threatening because the DNA damage, protein coagulation, everything of that sort happens, and that is where thermia is a physical therapy. The cells don't find a way to escape, and it is instant. Uh, so I will quickly move on. So beyond the cell-based, what we saw, then we implanted a tumor, we injected cells, the nanoparticle in them, and then we are triggering with our laser, which is, this laser is very mild laser. This is less than 1 milliwatt, so it do not have too much of a power. So it is safe for skin. But in the mixture with the nanomaterial, it immediately, you can see this tumor has now dehydrated. So it become whitish and more solid. That glow of that watery glow was gone. And next day it is a hemorrhoid. So the whole mass we could take out in a day, in a matter of 5 minutes of treatment, and within one day we see the effect. We have in other mouse, we have grown a bigger tumor, we cut it open, and then see that how depth was that. So we are trying to get a sense. So you can see the burn wound is something like on the periphery from the triggered side of the laser, because the photonic signal do not go very deep. So it is limited by few millimeters and so on. So the far end of the tumor may not be affected in one shot. So that's where the booster dose and so on could be required. And this was further confirmed through immunohistochemistry and so on, other study. So this brings to now, as I showed you, that our luminescence-based imaging can help us monitor this in a non-invasive manner over a period of time. And so in a controlled tumor, the tumor continued to grow and spread in metastasis is forming. And basically by 5 weeks, it's gone. I mean, we cannot, there is called ethics, animal ethics. So we have to sacrifice this mouse because it is very painful for that mouse. So we have to stop our experiment on day 35, around day 35. But if I do a treatment on somewhere around 22nd day of growth, the tumor reduced, uh, but it didn't vanish completely with a single dose. It regrow, but in the process, what happens is I could easily see that the mouse can now survive up to 70 days. So it is doubling the time. It's not adding 1 year time in a cancer patient life or a 5 years time or 10 years time. I'm doubling the time of the lifespan. That if untreated, in comparison to the untreated, I could now, if I do this treatment, the patient can survive double the time than was expected. So this was a definite rigid gain that we can achieve through just a single modality, that is the photothermal therapy. But what we are challenging further, we as a researcher, we don't stop. And because this is a new kitty, we have to, um, clinician will ask you 100 questions before it can be integrated. So we are not stopping. We have to know all of it. So what we are seeing is we can treat the primary and that expands the life expectancy, but we cannot stop metastasis. So the relapse will happen, continue. So now what to do? And that's where we are actually bringing, so these are the last couple of slides for me today. So a couple of other approach that we are developing in terms of making it more precision nanomedicine therapy. So there are many forms. So we are stuck with this photothermal, but there are also other forms of hypothermia, like magnetic hypothermia is very common. Ultrasound-based materials are also there. People are using radio frequency-based ablation are also there. What we are talking today is a biomimicry. So some of you may be Bengali, I don't know. I'm a Bengali. And there is a proverb called, we say, kaante se kaanta uthao. Hey, understood? Meaning kill the poison with the poison in a way. So what we are thinking is that, the cancer cells, they have receptors. And this is like we are vacating cancer cells. So a patient comes in, the idea is this, a patient comes in, the surgeon takes a surgery down, hand me a piece of tissue, which I will grind and make those cells, cancer cells, hollow. So only its membrane will remain. This is a preparation we can do. We have optimized in the lab. And so when you make a cell hollow, because the nucleus is gone, cytoplasmic mass is gone. So now it's like a RBC kind of shape it comes, because it's like a cup shape in transmission electron microscopic picture. So you can see, and then I can load them with my photothermal agents. And now these membranes are intact, and this is called homotypic interaction. Now I delivered this material, and the cancer cells, the growing tumor that is there, so it is like allogenic treatment. Okay, so these cells will go and find and bind to the remaining cancer mass, and then so photothermal agent is directly delivered there, accumulated, and now I just have to trigger it with my laser, and I can make the rest of it vanish. Uh, so this is one strategy. The other strategy, another student of mine is working, is again very, very provocating. It's immunotherapy is very, very well taken for cancer these days. CAR T cell therapy is something that you might have heard of. Um, so what we are also thinking that when I'm exposing cancer cells, triggering it with a photothermal therapy, so there are dams that will be expressed in the treated tumor cells. And then of course, most of the previous experiments that I have done is on immunocompromised mouse. But a cancer patient is generally not immunocompromised. We all have our immunity. So if we need to do more realistic experiment, thinking that we started this line that we have to characterize how the body's immune cell will react to that site where we are treating. Because after all, we are creating a burn wound. So there would be a lot of rush of T cells and macrophage, and you can think about it. And so that's what we have done, a lot of characterization. And finally, this part we are doing as a therapy, that it is triggering it is like it will work like a vaccine. The photothermal treated cancer cells that surface antigens, it will it will attract body's immune cells, and then these immune cells, they got adoptive therapy, and they will, if they find another cell somewhere, so treating the metastatic nodule in a distant cell. So that is what is being experimented here. The first tumor, which is on the left, it is being treated with the photothermal, and the second, how it is affecting the second tumor, that is being checked. So what we are seeing, that if we don't do anything, then metastatic spreads happen, and within a syngeneic mouse model, so this is very, very aggressive mouse breast cancer tumor. It spreads everywhere, and then we have to sack. But when we do implant two tumors, we see that even the second tumor is not being able to grow well. Okay, otherwise it is very aggressive. Within that 21 days, I have only treated the left side, not the right side. So why is the right side is suppressed is because the body's immune activation, and that is what is an approach that we can thinking of integrating, where photothermal followed by like a cancer vaccine, we can use this way. So this is the last piece of result I wanted to share, and that's all. I'm not summarizing; it's a popular talk. I'll be happy to take your questions and discuss further. So thank you everyone. Make sense?